In this video, we're going to look at the lipids that make up your cell membrane. And we call these classes of lipids glycerophospholipids. And these are glycerol 3-phosphate. So we have our glycerol here. At the third position, we have a phosphate. At the 1 and 2 position, again, we can have fatty acids. Now, this phosphate usually has another component attached to it, and based on what is attached to it, it gets a special name, such as phosphatidic acid, phosphatidylethylamine, and so on and so forth. And all these different types of head groups leads to lipids that have different functions in the cell membrane. Now, the way we know what these glycerophospholipids look like is through hydraulic reactions by enzymes called phospholipidases. So a phospholipidase is simply an enzyme that cuts off part of a glycerophospholipid. And one well studied is phospholipase A2, which cuts off the second fatty acid. And what you're left with is a lysophospholipid, which is shown here. Now, these lysophospholipids will disturb the cell membrane and eventually lyse the cell. And this is what bees and snakes actually use in their venom. They have these lysophospholipids which will disrupt their prey's membranes. These enzymes are actually well studied, as shown here is an image of phospholipase A2 working on a lipid. And what you can see is that the lipid's here, and you have a bunch of aromatic residues that are interacting with the fatty acid chain and holding it in place. And the reason why they're so interesting to study is that you have a protein where you're going to have hydrophilic residues on the outside. So the question is, how does this mainly nonpolar residue interact with an enzyme that's polar? And here we can start to look at some of the mechanism through that image. These lysophospholipidases can also act as a signal by having this phospholipase A2 cutting off. You can send what's left to a different cell, which can interact with that cell, and various messages can be transmitted through different cells in a tissue. Now let's look at plasmalogens. And plasmalogens have your phospho group, your glycerol, but this time at C1 we have an alpha-beta unsaturated ether linkage. And this linkage or this double bond is usually in the cis conformation. Now, your most common type of plasmalogens are ethylamine, which you would have at this X position, an ethylamine, your choline, and your serine, which is the side chain of serine. Now, we don't really know what plasmalogens actually do, but the idea is that they will react with oxygen-free radicals. Oxygen-free radicals are very deadly to a cell. They can cleave your DNA, or they can mess with proteins, and so we really want to take care of them. And one of the ideas is this plasmalogen will absorb the free radical. You could interact with this double bond, but if you interact with that double bond, it really doesn't matter. The cell is still staying alive. You're not messing with a protein, and you're not messing with DNA. Now, another type of glycerophospholipid is the sphingosine. And your sphingolipids, actually shown here, are actually the major membrane component. And here you have your glycerol and you have your NH3, and that is why it gets the name sphingosine, because it has this NH3 group. And then you have a double bond at position 3. Now you can modify the sphingosine even further, where this NH3 gets a fatty acid residue, and we call this a ceramide. So it's an N-acyl fatty acid derivative, but it is still a sphingolipid. It has the same type of backbone, you just get a fatty acid to call it a ceramide. Now let's look at sphingomyelins, or sphingophospholipid. So this is kind of a combination of the glycerophospholipid and the sphingosine, because we have our phospho right here, and this is actually phosphocholine, so here's choline. And then we have some fatty acids. So this is sphingomyelin. So it's either going to be phosphocholine or phosphoethylolamine. And 10 to 20% of the lipids in your lipid membrane are actually sphingomyelins. Now another type of sphingomyelin is cerebosides, Glycosphingolipids. And this is your head group with a sugar now. So the type of sugars or the type of cerebrosides you can have are galacto or gluco. Those are going to be your most common with lactoside and a glucose as your head groups. And these are non-ionic, so they're not charged. And you have your gangliosides. And these are very complex. So here's some gangliosides right here. As you can see, there's a lot going on with the different types of sugars. But the head group is usually a sialic acid. And these are also your cell surface membrane. And 6% of your brain lipids are gangliosides. So they have some function in the brain, but we're really not going to go into that all that much. And that's it for your cell membrane lipids. I will see you in the next video.